All right, let's get right into it. We've all heard that scientists sequence the human genome, right? Our entire genetic code? But here's the thing. Having the 3 billion letters of our DNA is one thing. Actually being able to read the book is a whole different ballgame. Today we're talking about a brand new AI that's finally starting to teach us the language of our own biology.
I mean, really think about that for a second. We have this gigantic 3 billion letter string of A's, C's, G's, and T's. But where do the genes actually start and stop? Which parts are the instructions, and which parts are the switches that turn those instructions on and off? It's like someone handed you an epic novel with all the letters jammed together. No spaces, no punctuation, no chapter breaks. That's the huge puzzle we're trying to solve.
And there's a name for this whole process. It's called genome annotation. This is the incredibly detailed work of going through and labeling everything. You're trying to find the genes, the promoters, the enhancers, all the critical components. It's literally like adding the punctuation and the chapter titles back into that book of life, turning raw data into something we can actually understand.
So here's what we're going to cover. First, we'll look at why reading the genome's language has been so tough. Then we'll see how a really cool idea from image-recognizing AI changed everything. We'll meet the new model, SegmentNT, see how well it actually performs, and then get into its mind-blowing ability to translate between species. And finally, we'll wrap up with what this all means for the future of science.
Okay, so let's set the scene. For years and years, actually making sense of genomes has been a massive bottleneck in biology. We got incredibly good at sequencing DNA really fast and cheap, but understanding what it all meant, that was still a slow, painful, and super difficult process. A big part of the problem was the tools we were using. You'd have one program that was decent at finding genes, and a completely different one for finding promoters, and another for something else. Each tool was its own little specialist, trained on a pretty small data set, and they just couldn't see how all the different parts of the genome worked together. It was slow, inefficient, and you basically had to start over for every new species you looked at.
So, to break through this, researchers really needed a totally new way of thinking. And the inspiration, amazingly, didn't come from biology at all. It came from computer vision, from the same kind of AI that helps power things like self-driving cars. The key idea is called semantic segmentation. Now, what that means is when an AI looks at a picture, it doesn't just draw a box and say, that's a car. No, it goes through and labels every single pixel. This pixel is road, this one is tree, this one is sky, this one is car door. It creates this incredibly detailed, color-coded map of the entire scene.
And that was the big light bulb moment. What if you could treat the genome like a one-dimensional image? What if you could train an AI to go through our DNA, base by base, and label every single one? This nucleotide is part of a gene. This one is part of a promoter. This one is an intron. You get a complete, high-resolution, functional map of our entire genome.
And this is where the hero of our story comes in. Meet SegmentNT. That stands for Segment Nucleotide Transformer. It's the AI model built to do exactly that, to read and segment the language of DNA down to a single-letter resolution.
Now, the secret sauce behind SegmentNT is something called a DNA foundation model. It's actually a pretty simple but really powerful concept. Before you teach the AI to do any specific task, you first just make it read. You have it absorb billions and billions of letters of DNA from all kinds of different species. It starts to learn the basic grammar, the common phrases, the fundamental patterns of life's code. Pretty much the same way a model like ChatGPT learns about human language by reading the whole internet.
So how does it work in practice? Well, first, you feed it a long chunk of DNA, up to 50,000 letters. The foundation model part then reads that sequence and figures out the biological context. It understands the grammar. After that, another piece, called the segmentation head, scans that data and identifies all the patterns at once, finally spitting out that complete, detailed functional map.
And when I say detailed, I mean it. Instead of having a bunch of different tools, SegmentNT identifies 14 different types of genomic elements all at the same time. We're talking genes, of course, but also regulatory parts like promoters and enhancers, splice sites, the whole shebang, all in one single pass. It's a massive leap forward.
Okay, so the idea is great, the architecture is cool, but does it actually work? The proof is in the pudding, right? The big question was, does that whole pre-training phase, making it learn the language of DNA first, really make a difference?
And the answer from the researchers was a huge, resounding yes. It turns out using that pre-trained foundation model was an absolute game changer. It helped SegmentNT learn the task 7 times faster and, in the end, its performance was twice as good as a model that started from scratch. This pretty much proves that learning the language first isn't just a nice-to-have, it's essential.
And you don't even have to take my word for it, you can see the difference right here. On the left, that's the performance of SegmentNT with its pre-trained brain. On the right, that's a model with the exact same structure, but without that pre-training. I mean, the performance gap is just massive. It's not a small tweak, it's a completely different level of understanding.
So how does it stack up against the old champions? Well, they put it head-to-head with a tool called Augustus, which has been the gold standard for finding genes for a long time. And SegmentNT came out on top. It was better at finding the genes that were really there, that's what we call higher recall, and it made fewer mistakes by flagging things that weren't actually genes. That's higher precision. It just did a better job.
But this is where the story gets really, really interesting. This is where things get kind of mind-blowing. The researchers started to test just how far the model could stretch its knowledge, and what they found suggests this isn't just a tool for reading human DNA. It's way more powerful than that.
So get this, the very first version of the model, it was trained only on the human genome. That's it. But then, just to see what would happen, they gave it DNA from other primates, like gorillas and macaques. And it worked. It could already find their genes with pretty high accuracy. That tells you it didn't just memorize human DNA, it actually learned some of the fundamental rules of how mammalian genetics works.
So, naturally, they pushed it even further. They built a new multi-species model, this time training it on DNA from humans and 5 other animals. We're talking mice, chickens, even flies. And this new, more worldly model got way, way better at annotating the genomes of animals it had never seen before. It was like the more languages it learned, the better a universal translator it became.
And that brings us to the most incredible result of the whole study, the ultimate test. They took their multi-species model, which remember had only ever seen animal DNA in its entire life, and they gave it the genome of a plant, something totally alien to it. And it worked. Unbelievably, it worked. It accurately found the genes in plants like corn and rice. It had crossed the kingdom divide. It had somehow learned a truly universal language of life. So you can see this isn't just a cool academic paper. Having a tool that can map out pretty much any genome this quickly and this accurately, well, it has some really profound implications for medicine and biology.
So why does this all matter? Well, for one, we can analyze the messy, scrambled-up genomes of cancer cells way more effectively. We can finally figure out what a tiny, disease-causing mutation actually does because we can see if it breaks a gene or messes with a switch or hits some other important spot. And for science as a whole, we can now rapidly map out the DNA of newly discovered species, unlocking their secrets and building this incredibly rich, detailed picture of how life on Earth evolved.
It really feels like for the first time we have a tool that is starting to read the book of life with real fluency. It's beginning to understand the deep grammar that connects not just you and me, but humans and animals and even plants. We've moved from just spelling out the letters to actually understanding the sentences. And that leaves us with a pretty exciting question. What are we going to discover in the next chapter?